morning, boys and girls. Okay, we are going to start talking about skin today. Okay, and in the lab, you will see skin slides next week. Skin is our leather, guys. It has other names as well: cutaneous membrane or integumentary system. Okay, and believe it or not, this is the largest organ system with largest weight and largest surface. Kind of, if you don't think about it, you would never know, right? These are your objectives about the epidermis and the dermis. Today we are going to focus on the epidermis. Okay, the functions of the skin. Of course, number one is first line of defense, which means protecting us from the outside world from many things. What are those? Protection from physical, mechanical friction, including your thick skin and your nail, including our hair, actually. Everything protection. It protects us from chemical agents, from biological bacterial invasion, meaning infections, right? And the fast repair of the skin protects you from further bacterial invasion into your body, into your blood. It also protects you from water loss or dehydration. It also protects you from wetting, meaning you can take a shower or you can take a bath, jacuzzi, and the water doesn't go into your body. Okay? It also protects you from the UV light of the sun by synthesizing melanin protein. And when the melanin is increasing, that's what we do tanning. And you know, you can do tanning salons and you can do it artificially as well, not only by sunshine. Okay, so it also protects you from heat and cold, and skin is a major player in thermoregulation of our body, okay, with many different structures, including the blood circulations, including sweat, with another word, sudor, including hair. In animals, it's called fur. And you can probably say in animals, it's probably hair. It's more significant than in humans. And fat insulation in the hypodermis is also contributes to thermoregulation. The skin also protects you from dangerous stimuli. It is our largest sensory organ. So you can think about skin as part of the sensory nervous system. Why? Because it's going to have a lot of touch, pressure, pain, temperature receptors. Okay, and function number two, skin is also an endocrine organ in a sense that this is where synthesis of vitamin D3 occurs. The first step of vitamin D synthesis and activation. So the precursor is a cholesterol molecule, and the UV light turns the cholesterol into inactive vitamin D3. And after this, the vitamin D goes into the blood and needs to be activated. And I believe you guys already learned that, right? No? Maybe I will add it later to the PowerPoint that you have it. It's going to be activated first in the liver, then it's going to go into the kidney. And where is it going to go ultimately? After fully activated vitamin D, will go to the small intestine and helps the calcium absorption into the blood. So that's the way vitamin D is associated with calcification or strong bones. Okay, so you guys want me to add this here? Okay. And also, what is very important in the clinic, guys, that when you see a patient walks into your clinic, the first thing you're gonna see their condition and the color of the skin. That is immediately is going to give you an idea what type of disease the patient may have. Okay, the color, the hydration, how the nails look like, how the hair looks like. Okay, it's all associated, just giving an idea about the condition of the patient. Okay, so let's talk about structure. On the left side, you see kind of like a model picture, and you can see the layers of the skin. And then analogous on the right side, histology. Okay, the good news is, guys, that you already saw this slide. So thank God it's already from foundation block. There is a little bit of, you already have a little base knowledge. Okay, but anyway, let's talk about the layers of the skin. The brown on the top is what we are going to call epidermis. Okay, the pink under it, you see much thicker layer is the dermis. And the yellow under the pink is the hypodermis, or also some people call it subcutis. Okay, so how does this look like on histology image? How are you going to see this in the lab, right? With hematoxylin eosin. The purple structure on the top is the epidermis. Then this really large territory pink is the dermis. And the dermis is going to actually have two layers. The papillary dermis which is right under the epidermis. And the bigger part of the dermis is the reticulodermis. But as I said, we're going to really cover these guys next time. And what is the major component of subcutis and hypodermis? Fat. Adipose tissue, adipocytes. Okay, and I mean, I labeled here for you because it's already here that you can see some ducts running in the tissue. This would be a sweat gland, and the sweat gland duct as the sweat is going upwards and watering the surface of the epidermis that will cool you off by evaporation. Okay. Let's look at, this is what you already know, actually. The name of this is the new thing, that the name of the layer, and you can say this is kind of your anatomical name, is your epidermis. What that really means is on the top of the dermis, because epi means on the top. This is basically the only new thing here, right, today. The rest you actually already know. What type of tissue is sitting in the epidermis? It's an epithelium. What kind of epithelium is sitting in the epidermis? This mouthful, right? Stratified squamous epithelium with keratin, which can be thin keratin or thick keratin. And this is where I would like to give you a warning for OSPE that the previous OSPE, you had one question about this issue, and only like 5 students remember that we have actually 3 stratified squamous epithelium based on the keratinization. No keratin, thin keratin, thick keratin. So please don't remember saying that in your answer. What now we are doing cells, right? The living units. What kind of cells are sitting in the epidermis? Epithelial cells. How many kind of epithelial cells are out there? Squamous epithelial cell, tubular epithelial cell, and columnar epithelial cells. It's so simple that it's hard to remember. Because you are thinking about a very difficult name. No difficult name. And you don't put there simple, because the moment you say simple, you are talking about already a tissue where the cells are sitting in one layer. But what you're gonna see is a mixture of squamous, cuboidal, and columnar epithelial cells. They are all there in the epidermis. And because these cells are very special and their major function is synthesizing keratin protein, these epithelial cells, all of them, doesn't matter squamous or cuboid or columnar, together their, so to speak, functional name is going to be keratinocytes. You could say the squamous, cuboid, columnar are their structural name or more generally. Okay? So they are all keratinocytes because they synthesize keratin protein. Okay, so this is the slide what I call the structure slide. So I'm analyzing every single layer. So we are analyzing all the sublayers and the cells of the epidermis. Okay, we have 4 sublayers in the epidermis, and first we are going to look at the thin skin. Okay, and we are going to start in stratum basale. And we're going to start moving upward. And the reason is because stratum basale is germinativum, where the tissue is actually growing. And then the tissue is going to be pushed up and up and up as more and more mitosis occurs in the basal layer. Okay, so let's talk about, so I like to do usually not just here, but in other places, put structures in order of physiology. And that's what we are doing. So stratum basale or germinativum is where the epidermis is growing. Germinativum means grow. What do you see here? One layer of stem cells of the keratinocytes. Okay? And actually, they look like columnar epithelial cells with oval nuclei. And you remember, if it's oval, it can be a mixture of oval and round nuclei. Depends on the cup. But the nucleus is oval. Okay? So basically, you see one layer of columnar epithelial cells here. Then let's move upward. And this middle part, which is the thickest part, okay, is called the stratum spinosum or prickle cell layer. This is not one layer, this is multi-layers of keratinocytes. And how do they look like? You see everybody has kind of a roundish nuclei. So we're gonna call them cuboidal epithelial cells. Between the between the cells at the plasma membrane, you're gonna see a lot of desmosomes that they look like prickles. You can see kind of a little bit already on this image, but I will show you later even a better one. Okay, so many desmosomes are holding these keratinocytes attached to each other. Okay, now we are moving higher and we are hitting the stratum granulosum. So watch the name granule. Why is it called stratum granulosum? Because keratohyalin granules, or you can just say keratin granules, develop by this time as the keratin synthesis is occurring. You will see that on the next slide. Okay, so this is about 2 layers of keratinocytes or granular cells. Okay, so how do they look like? Flat epithelial cells with flat nuclei and basal granules in them. You can see that actually very nicely in the microscope when you zoom in. Right now, all you see is that it's darker, right? But the darkness is coming from the granules. Okay, and finally on the top, do you see it looks like this basket looking place? This is what called orthokeratin. Very typical of usually thin keratin organization in the stratum corneum, which means literally horny layer. Not, you believe it or not, now you are looking at a non-living layer or a dead layer of keratinocytes. So when you look at carefully, can you see nuclei? No nuclei. Cytoplasm are dried up, organelles dried up. Of course, organelles are not visible in light microscope, but if you would use electron microscope, you would tell that the cell is dead. Yeah, remember we just saw the dead dying cartilage and the epiphyseal plate, right? So the nuclei again and the organelles are going to degenerate. One thing again that you cannot see on the light microscope, but there is some lipid covering between the corneocytes. So these dead keratinocytes has a new name, corneocytes, okay? And they are covered with a lipidy structure, lipidy molecules to prevent water loss, okay? All right, so let's move on to the next slide, which I call the more like the physiology kind of slide. This was your structure slide to analyze all the structures. This is what's happening in the different layers. The keratinocyte, and this, you remember, start with the title every time you look at an image, because it tells you that keratinocytes in the epidermis undergo a programmed cell death called cornification. Okay, the oxygen diffuses up from the capillaries of the papillary layer of the dermis. I really don't like this thing coming down on and off, but what am I gonna do? Anyway, the oxygen is diffusing from the capillaries of the papillary layer of the dermis to the keratinocytes to give them oxygen and nutrient supply. And as the oxygen and the nutrient, so basically, let me show you, this is what we are talking about. This pink territory here is your dermis. This is where the blood capillaries are. Yeah, sometimes we call this underlying connective tissue. I hope you remember that. And it's actually areolar connective tissue, but that's not the purpose of today. This is where the capillaries are, and the oxygen is going to start moving, diffusing upward to keep the keratinocytes alive. And as the oxygen, where are the more oxygen? Obviously at the basal layer, and technically those guys need the oxygen the most because they are doing mitosis, right? And then after that comes the oxygen less and less and less, contributing to the death of the keratinocytes. And there are other factors here that we're not going to get into in this level. Okay, so let's do again from bottom up. The stratum basale or the germinativum, as we said, this is where the growth occurs. This is where non-stop mitosis occurs, or with another word, proliferation. So you guys remember you have a parent cell, and then it's going to go through mitosis. You end up with two daughter cells. One of them remains stem cell, the other one says goodbye, bye mommy, and start moving upward. Okay, so that's why if you look at the strat, so that's why the whole tissue is kind of goes up a little bit. That's why I put it here for you, push up. Okay, so this is where the upward migration begins. And there's another change occurs. These cells do not do mitosis anymore. Okay, the mitosis is only in the stratum basale. This is what we call they exit the cell cycle, but at the same time, keratin synthesis also begins. Okay, because that's what these guys are doing all day long. Okay, so another push. Okay, and we are in the stratum granulosum now, where cell death actually begins. And remember, you will see keratin granules. Then push further up, and in the dead layer, what we already know as stratum corneum, is where full keratinization is visible. And what you see is keratin squams. Squam is like the scale of the fish, like that type of structures. And what is going to happen as the layers are pushed and pushed and pushed, ultimately the dead layers will peel off and falling off from the surface. That's what we're going to call desquamation. Okay? And the falling off dead skin often is dandruff. I'm sure you hear is, you know, that when they're selling the shampoo. And you believe it or not, that's majority of the dust in our houses, except in Saudi Arabia. Here is the Sahara is sitting on your table. In the rest of the world, dandruff is sitting on the table. Okay, so let's look at the next image. So this is more looking into the cells, but just a little bit more information. So this is your columnar basal cell. Then this is the spinous cell, more cuboid, more squished. So you have two spinous cells next to each other. You can see very nicely the desmosomes are connecting at this point. And then here is your granular cell. And this is your cornified dead cell. Okay. And as we discussed, the oxygen is decreasing. And this is what's happening inside. The basal cell actually starts synthesizing keratin tonofilaments that becomes tonofibrils in the spinous layer, keratin granules in the granular layer, and keratin squams in the top layer. Okay, and again, I remind you that we did this before in the muscles, right? When you say filament or fibril, what's the difference? The size. The word gets longer, the structure gets smaller. Filament is a smaller fiber than a fibril, and the fibril is a smaller fiber than a fiber. That's the way the terminology goes. Okay. So let's look at, this is, as I told you earlier, this is a better image to show you the desmosomes. I had to borrow, this is borrowed from our histology book, Wheaton. So stratum spinosum in light microscope is very successful. Actually, in some of our slides, you should be able to see as well that you see many layers of cuboidal epithelial cells. The desmosome, do you see these lines, blue lines between two keratinocytes plasma membrane, between two plasma membranes? Those are the prickles or spines. That's why they decided to call this spinous layer. And yes, it does look like a pavement. And yes, brown nuclei. Okay, so how does that, and this is again, repeat, we did this before, right? How does desmosome look like in electron microscopy? So here is going to be plasma membrane 1, plasma membrane 2 of keratinocyte 1, keratinocyte 2. And this little drawing that created, and I borrowed it from Wikipedia here, is very helpful to, so the blue would be the two plasma membrane. Then on the blue, there is this red protein plaque, right? And then from between the two plasma membrane, you have adhesion proteins, cathedrin, and from the red plaques, you have, guess what? Keratin, right? That's what these cells have anyway, right? So here are going to be the keratin filaments. Okay, which is going to be part of the cytoskeleton as well of the cell. And you guys remember we talked about cytoskeleton before, and we went through a few of them, and I said, which one is the one that can be intracellular cytoskeleton, and it can be also big, large, extracellular protein fiber. Which one it was? Here we go. Okay. Right now I'm showing it as a cytoskeleton. When we were making the falling off keratin from the surface, that was extracellular keratin. Okay? One more thing, I just wanted you to picture it in your mind, okay? That so who is really protecting you guys from the outside world? A dead layer of cells. Between you and the outside world, there are dead cells, and they are the one who protects you. Just give a little bit of thought to that. Okay, now this image is going to give you the light microscopic and electron microscopic image of the stratum corneum or keratin squamous. This is the way you're going to see it in light microscope. It is a thick skin. So this is all keratin, dead keratinocytes. And this is the way the squams are going to look like, as I told you before, like scales falling off. Looks on electron microscope. Okay. Okay, protection of epidermis by glands. Now this actually, I touch on it here, but we're gonna get into more detail of the glands when we are doing dermis, because glands are actually sitting in the dermis. What you're gonna see is that the ducts has to go through the epidermis. But anyway, this is you see how white looks like fat? Well, it is fat, okay? Because this is the sebaceous gland or oil gland that makes fat and is going to moisturize the epidermis, okay? And the other one is the sudoriferous gland or sweat gland, and this is the fellow that is going to be involved cooling the epidermis and cooling us, okay? But as I said, we are coming back to these guys. Let's compare the thick and thin skin with each other. And again, I just want to make sure that you understand that the thick skin keratin is so thick that it's thicker than the epidermis, or the whole thing is the epidermis, but where the living cells are. Okay, this whole thing is the epithelium, guys. So this would be the basal layer, right, going up and down in a wavy line. Okay, then this bigger, and you see the nuclei everywhere, the blue nuclei. Then here are the bunch of round nuclei in the middle. That's going to be your stratum spinosum. Then do you see the dark purple? That's the stratum granulosum. Then I put an L here for you. It looks a little bit more solid line here. Could be better. Some slides you see better than the other. This is the new layer one can see in thick skin, the stratum lucidum. So you remember we had 4 sublayers in the thin skin. So in thick skin, we're gonna have 5 sublayers, which is the new guy, the stratum lucidum, right? You could say under the stratum corneum or between the stratum granulosum and the stratum corneum. Okay, so, and now you can see very nicely also the collagen fibers of the dermis. Okay, so let's move now to the thin skin. And again, please remember this entire thing is your keratinocytes from all the way. I just noticed that sometimes it's confusing. That whole thing as it goes is the epithelium, the keratinocytes. And look at the thin keratin on the top. Again, looks like a little basket, right? And you can see very nicely the stratum basale. It's darker. Then the whole pink thing is the spinosome, and then this is again a little darker, but not as good as on this side, is the granulosome, and then comes the thin stratum corneum. And this guy is, again, you see the size? In thin skin, the stratum corneum is much thinner than the rest of the epidermis. Here, the stratum corneum is much bigger than the rest of the epidermis. There is one more difference here, guys, the hair follicle. In thick skin, there are no hair follicles. In thin skin, everywhere hair follicles. And I put here for you a follicle with an actual hair. And one more thing, where are they located? Thick skin is always only two locations, hands and feet. I put the sole and palm, that's it, okay? Those are the, why do I need thick skin cell? Because these are the parts of your body that has the most friction, right? And everywhere else, actually, there is thin epidermis. Okay, I just labeled it for you all over again for not to be confused that stratum basale, stratum spinosum, stratum granulosum, lucidum, corneum, outside. And here is your dead layer between you and the outside body. Okay, so this is just a repeat. Okay, now we're going to start talking about the other cell types exist in the epidermis. Okay, so what did we talk about so far? The keratinocytes. They are the majority in the epidermis, but here and there you will find some scattered cells, Langerhans, melanocytes, and Merkel cell. You can see them actually very nicely. Everything, anyway, orange is the Langerhans, and you can already see the Merkel and the melanocytes are going to be in the stratum basale. Okay, so this is repeat again. Just remind you that this is your cell one. What is cell one? Keratinocytes. What are they doing all day long? Keratin. And they are the majority. Technically, right now, I don't see any other type of cell here. Only keratinocytes. Okay, now this slide, maybe I could have put it somewhere else, but either way, I wanted to just touch on it again on the oxygen supply. And you can see very nicely the capillaries. These are all capillaries in the... Okay, you can see there on this slide, you can see very nicely the capillaries in the underlying connective tissue. And the arrows are showing you how the oxygen is going to diffuse from the capillaries to the keratinocytes. Okay, and I also put here for you that the underlying connective tissue is areolar or loose irregular connective tissue. I cannot hear you, sweetheart. This is right under the epidermis. And when we talk about dermis next time, we're gonna do it again. Okay? So now comes cell 2. Okay? We overanalyzed keratinocytes, but I wanted to remind you again that that's your number one. Number 2 is the Langerhans cells, or LC, which is an immune cell in the epidermis, about 3% of the cells. And some books call this, or some actually probably I got it from this NIH paper, they call it high-level clear cells. Why? Because they are higher located. Usually you're going to see them in the stratum spinosum. Okay, let's look at the origin. This is, guys, going to be a macrophage, guys. So where do macrophage come from? Just like osteoclast, from the hematopoietic stem cells from the bone marrow. Okay, and if it's a macrophage, technically It can be anywhere because it can move to the place of infection. But usually you will see it on a higher suprabasal layer. This is the fellow who discovered it, named after him, German guy. Okay, so here is a Langerhans cell buried in the stratum spinosum. You can see branches, cytoplasmic processes, and it looks like a little eye looking at you. When you look at the blue, purplish keratinocytes, suddenly somebody is looking at you. Okay? There's a clear hollow around the nucleus, which means the cytoplasm doesn't stay with hematoxylase. Okay? There's a famous organelle here that we call Bird-back granules inside the cytoplasm that you can see only on electron microscopy. Okay, so we did the location, function, immune cell, first line of defense, antigen presenting cell. We did that actually also before. That after it, the processes are monitoring for new antigens, bacteria. After stimulated, they migrate first to the dermis, then to lymph nodes, and then they will wake up the cell-mediated immune response. I know that this sounds Chinese right now because you didn't learn immune system yet, but this is what macrophages do, okay? Okay, so this is a picture of a Langerhans cells, and in this is the nucleus, and these little fellows that I put in the zoom sign is the Bird-back granule. When you magnify the Bird-back granule, that's when it's gonna look like a tennis racket. On one side, it looks like a vesicle. On the other side, it looks like a striated rod. And people theorizing that this is the structure that is involved in antigen processing. And this NIH paper actually talks to you about the fact that the Langerhans cells are targeted by HIV infection. In fact, once the HIV goes in through a receptor called Langerhans receptor, once it goes in, it will be internalized in the Bird-back granules. Okay, so of course, if it's an immune cell, the question is when their number increase and when their number decrease. Of course, they increase if you have inflammation, if you have allergic reactions, you have transplantation. This is the cell is gonna reject the transplant if blah blah blah, and also number increase if there is a tumor in the skin. So Langerhans cell is trying to get rid of the tumor. When is the Langerhans cell decrease? UV light can kill it, carcinogen can kill it, immunosuppression, medication can kill it. So therefore, it's gonna protect you less from tumors. Okay, so now I'm coming cell number 3. And that's going to be, guys, your melanocyte. And this is called low-level clear cells in the stratum basale. Whatever you need to know about melanocytes, guys, is all summarized here. It's also a branch cells with dendrites. You can't really see it in microscope. 2 functions: melanogenesis, it makes melanin from tyrosine. Number 2, transfer the melanosomes into the keratinocyte. So guys, once the melanin is made in the melanosomes, they do not stay in the melanocytes. They're gonna go over to the keratinocytes for protection. I will show you in the next image. They are in the stratum basale. That's why they're called low-level clear cells. They start from the neuron crest. 2 to 10% of the basal cells. And there are 2 kinds of melanin, black, brown, and red, yellow. And we already did that. I'm going to show you an image about the transport of melanin from the melanocytes into the keratinocytes. Who wakes up these cells, guys? UV light, tanning, and it has a hormone too, melanocyte stimulating hormone from the anterior pituitary. And epidermal melanin unit means that one melanocyte, you see the processes associated with the keratinocytes, he is responsible to protect. Okay, now let's look at the next image. And here, this is the way you're gonna see it. Again, you're gonna see an eye. The clear, halo around the nucleus. As I said, the branching doesn't show. Okay, and we actually covered everything, except I want to show you how the keratinocyte, how the melanin protects the keratinocytes. Here we go. On this image, do you see this blue melanin? And if you remember from introductory unit, melanin was one of those pigment molecules that had its own color, no need for staining. Look what they are doing. Which way is the sun is coming from, guys? From the top, right? So which way is it gonna protect these nuclei? From the sunny side. That's what they do. They are protecting the nucleus of the keratinocytes on the sunny side. Okay, I think there is another image of this. That this is just another how to say, special stain slide that you can see the processes of the melanocytes. And on this one, you can see the unit that one melanocyte and all the keratinocytes he is taking care of. Okay, so here's your melanosomes where the melanin is, and look, it is going to go over to into the exocytosis from the melanocytes and endocytosis into the keratinocytes. And where is it gonna be? Covering the nucleus on the sunny side. Okay, now this is another organelle in the melanocyte that we call melanosome, and it looks striated. Another organelle, cylindrical electrodense structures. And this is involved, basically, this purple structure, what I show you here on the right, is what you see on the left with electron microscope. Okay, now this is a very good image to stain the Langerhans and the melanocytes, because both are, you remember, on with a hematoxylation, they're gonna look at you like an eye. So how are you gonna determine who is who? The higher one is the Langerhans, the lower one is the melanocyte. But this one is a special stain. So the orange one is gonna be the higher, the Langerhans, and the pink is the melanocyte. And again, the special stain shows you also the processes that you're not gonna see in the light microscope. Now the cell number 4, guys, is the Merkel cell. This again is in all of these cells. Interestingly, all of the 3 cells, not the keratinocytes, has kind of processes. Okay, this orange fellow is the Merkel cell. The purple all around it are the keratinocytes. So again, we find another cell in the stratum basale. This is a unicellular sensory receptor. And again, we're gonna do this again when we talk about the sensory receptors, but we need to touch on it because this is the only sensory receptor. You have to be careful here because free nerve endings can be here too, but basically as a cell, this is the only sensory receptor exists in the epidermis. Most of your receptor organs are gonna be in the dermis. They are doing general sensation, and as again, stratum basale and stratum germinativum is the location. I also wanted to show you that between the keratinocytes and the mucosa, do you see the little lines? It is glued with desmosomes. Okay, so again, you're gonna see a clear halo around the nucleus, so another eye is looking at it. So what does that mean, guys? It means, where did I put this? Maybe on another side, but basically, melanocyte is also here. So on our slides, we cannot tell which one is the melanocyte and which one is the mucosa, because they both gonna have a clear halo around the nucleus, or what I usually say that somebody is looking at you. Okay? Mainly in the thick skin, palms, and soles. Okay? So let's talk about the function of the mucosa. This is a slowly adapting mechanoreceptor for gentle touch. Slowly adapting means that it's the best you understand adaptation is when you remember when you put on the watch and you feel the watch for 2 minutes, and after 2 minutes, you don't feel anything, and you can lose your watch because you don't feel anything. So this guy is slowly adapting, so it can feel longer. It can feel longer. That's why it is good for sustained touch and prolonged pressure. What you also see on the image that this cell, look, there's a chemical synapse with the axon of a sensory neuron that ultimately is going where? The dorsal root ganglion. Okay. Okay, now I summarized on this image for you guys the desmosomes. We saw desmosome between keratinocytes, right? Langerhans cells has no desmosome. Why? Well, remember their macrophages, they have to be able to move around. So you cannot bind them or glue them. Melanocytes also has no desmosomes, but Merkel cell, between Merkel cell and keratinocytes that I labeled here for you, also has desmosomes. Okay. So again, I repeat it here for you that when you come in the, how you're gonna see these guys in the lab, they like an eye looking at you. Okay. So here we go. You see, this is the way it's gonna see. This is the way you're gonna see it in our lab. Okay, so here is the epidermis, stratum corneum, one order. I'm asking for the cells. What is the cell? 1, 2, 3, 4. One is the all keratinocytes. And then now comes the eye, okay? Number 2, little bit higher, not in the stratum basale, has to be the high clear cell, the Langerhans. Then here, we can argue about which one is the melanocyte and which one is the Merkel cell. Okay? In the stratum basale. And then you guys can have a little bit of practice with this trivia at the end, okay? That you know everything what was covered in the lecture. The end. Attendance, please. Please get ready with your telephone.